Welcome back, everyone. Next up is Bioxytran Inc. They're on the OTCQB, trading under the ticker symbol BIXT. The company is a clinical stage biotech with two revolutionary platform technologies. They have an oral glycovirology drug that recently completed clinical trials and achieved 100% viral reduction in 7 days, a feat only accomplished by one other drug in the past decade. Their drug is a galectin inhibitor that neutralizes the COVID-19 virus and prevents it from entering cells. And they also have another platform drug being developed to treat strokes. Due to the molecule's small size, it has the ability to carry oxygen into areas of poor circulation. Please welcome Mike Sheik, Executive Vice President and Business Development. Nice to see you, Mike.
Yeah, nice to see you too, Anna.
All right, always a pleasure to have you on. So the presentation time is yours, and when you're ready for questions, let me know.
All right, sounds good. Well, I want to start by thanking everyone for being here. We're going to talk about next generation broad spectrum antivirals. What I want to do is I want to change the narrative away from people thinking that we're a COVID company. We're using COVID to show that we have a next generation broad spectrum antiviral. So here we are with our forward-looking statements. So you can read them at your leisure. But now what I want to talk about is the big picture. The big picture is that we are reshaping drug development. And I'm going to use the analogy of a dandelion. Okay, so I want everyone to get a picture of a dandelion. I don't know if you see on the right-hand side, I kind of have a dandelion superimposed in there where I talk about examples of the disease, not just the symptoms. So what everyone else out there is doing, and this is how we're different, is they're going after symptomatic therapies. They are trying to reduce the symptoms. So you have a disease and you're trying to reduce the symptoms. And that's, probably 90% of Big Pharma's focus is talking about symptomatic therapy. What our goal is different, and this is how we stand out from the pack, is we're going after disease modification. And you can see in there at the bottom, we're trying to pull the roots out, the roots of the disease out. We're not just trying to rip off the top of the dandelion. When you take away the symptoms being, the imagery being the dandelion, we're just ripping the top out of the dandelion. But the disease still remains, and what happens is it comes right back. So now let me give you some examples, okay? When we take opiates, for example, we're trying to treat osteoarthritis. So when you take the opiates, you treat the pain, but the osteoarthritis doesn't go away. It's still there. And how about Tylenol? When we take Tylenol, what we're trying to do is we're trying to reduce, they're reducing the fever, right? But what are they doing to the virus? Nothing. Now, how about eye drops when you have viral conjunctivitis? It soothes you, it makes your eyes feel better, but do you get rid of the conjunctivitis? No. And then now let's take another case. What about tumors? When you remove a tumor, does it stop the spread of the cancer? You see, these are all good examples. And I'm going to end with one final example that a lot of people take today, statins. They lower cholesterol, right? But does it scrub out the plaque that's in your arteries? So that's the difference between symptomatic therapy and disease modification. So now, what are we doing? What's our area of focus? Well, we have three areas of focus. We have two basically platform technologies. One is our glycovirology. You can see that at the bottom. And then hypoxia and degenerative diseases. That's our second pipeline. Now, what's the purpose of what we're doing here? Well, what we're hoping to do is we're hoping to eliminate viruses with our glycovirology platform. And then with our other platform, we're hoping to prolong lifespans and do disease modification on all these chronic diseases that are out there. Now, how do we do this? We're using what we're doing is we're targeting the disease pathogenesis. We're targeting this, what we call a galectin signature. So the signature of the disease using galectin antagonists. That's what we're doing. We're targeting that axis. And finally, what we're doing is we're using our broad spectrum antivirals, which are basically galectin antagonists, or in the other case, we're going to be using our acellular oxygen carrier. And later on in the presentation, I'll get into what that means. Basically, what it means is we have a molecule that just circulates blood through the body constantly. So how big is this market? So we set out this grand vision. So how big is it? Look at some of these indications. We have a massive pipeline potential. And we have a couple of molecules over here. So you'll see on the left-hand side, it starts with the molecules in the therapeutic area, the number of people infected globally, and then the market size. Now, look at the hundreds of millions of people, billion people for influenza, hundreds of millions in long COVID, millions in IPF, which is idiopathic pulmonary fibrosis. That's the last one on the list. NASH, stroke, cancer metastasis. These are multi-billion-dollar indications, but I'm fudging. They're 10 plus type billion-dollar indications. So what is our goal? Our goal is to get one drug, get one indication approved, and then it evolves into this platform technology. We honestly believe that we have something that could be bigger than possibly even Merck's Keytruda drug platform. You know, they have at least 15, 16 indications of cancer. They're doing, a couple years ago, they were doing, that's a dated number, around 14.3 billion in annual sales. How do investors make money in a biotech? So I'm going to do a little bit of education here. We do it through value creation. That means we have to advance our pipeline. So what I did was I showed you, if you look to the right-hand side of the slide, you're going to see our pipeline chart. What we need to do there is we need to keep moving to the right, keep developing and increasing our phase of development until we finally reach phase 3 and approval. That's what we're going to do. Now, how do we do that? We do that through licensing, we do that through partnerships, or we divest one of our product lines. So that's how we create value in our company. And that's how you, the investor, are going to see an increase in your share price. Now, who are we going to do this with? Well, that's on my next slide. We are going after big pharma. All the top big pharmas are listed there. You'll see below the iceberg area. They're in alphabetical orders. And what we did was we put the name of the company, and then we put what their specialties were. And then our specialties are on the top of the iceberg. Those are our therapeutic areas. And using our, obviously our platform drugs. So look at how it completely meshes with what big pharma is looking for. So these are our customers. This is who we're going after. Now, a little bit about the company. As an OTC investor, it's always nice to know what do we look like? What's our market cap? Our market cap is 23.5 million. There's 300 million authorized shares. All this is available on OTC markets, but I'm just giving you an overview. And then we have around 25 million in the float. And that's really an overview. Now, our market cap, we were really proud of our market cap. It's come down a little bit, but not that long ago, we were one of the few biotechs that hasn't suffered over a 90% drop in market cap. There was an article out there that I saw that showed that the average biotech from its high was down 95%. Well, the truth is, this is a value point to buy. We think we're were very low in terms of valuation compared to historical. And we think that's going to change and very shortly. Now, when it comes to biotechs and biotech investments, one of the key elements is the leadership and the management team. I'm going to focus, I didn't put up the entire management team, but I'm going to focus on our key scientist. This is Dr. Platt. You'll see here, right at the top, he had the first, we'll start right in the middle. He's the guy that coined the name galectin. So we're talking about galectin science. Well, he's the guy that coined the name galectin in 1992. And you'll see the journal article, a snippet of it right below. And then above that, the first article in the field of research of galectins, well, that was his too. From these founding type articles came 4,000 journal articles that all talk about galectins and how great of a target they are for receptors. So now Dr. Platt's been at this for 40 years in the research of galectins and carbohydrate binding proteins. Now you'll see right below his picture, he's written two textbooks. He's literally written the book on how to design carbohydrate drugs, which is what we're doing. He's run a bunch of public companies, journal articles, patents, clinical trials, animal experiments, you name it. When it comes to the right guy for galectin science, Dr. Platt, he's leading our team. So let's move on to the IP. Our IP, we have IP. We're continuing to develop the IP, and we're going to get more IP as we progress. I mean, there's not much to say about it other than that. So now let's talk about the ProLectin-M program. ProLectin-M, it's our oral antiviral drug. It's a chewable tablet. You take it, you feel better pretty quick. And with COVID, we've seen it turn PCR negative very quickly. So what's the problem out there? Look, we all know that COVID's a problem, but I'm talking in general. Set aside COVID, there's a lot of viruses running out there. We don't have effective treatments for viruses. So what I did was I put up a bunch of viruses here. COVID, influenza, shingles, RSV, viral conjunctivitis, long COVID. There's no effective treatment for these, but look at some of the numbers. 103 million for COVID-19, 26 million for influenza. And this is US data. 2 million for shingles, 2.1 million for RSV. I mean, look at the millions of people affected with viruses in the US. And now what happens is there's 2,600 RNA viruses that have been identified, but there's no broad spectrum solution to viruses. We have it for bacterias, right? We have this thing called the amoxicillin. It's a broad spectrum antibiotic, not antiviral, antibiotic. And then for viruses, one of the things that we could do is have a vaccine and try to prevent the viruses. But what we've seen with COVID is that vaccines really can't keep up with the pace that viruses mutate. A mutation is something that we'll talk about next. So ProLectin-M, one of the things that we're so proud of, we believe it's eventually going to be the first line of defense for all mutations of coronavirus. So what we have here is we have a versatile drug. And it doesn't care what sort of mutation or variant you have. It's fast. We believe, we like to call it sterilization. It means you're PCR negative in about 5 days. We don't think there's going to be any limitations on who's going to be able to use it. So if you're young, a baby, or if you're pregnant, or if you're older, we don't believe that there's going to be any limitations to its use. Right now, we're just doing it on standard risk patients, but we'll expand that. And then it's efficient. We're trying to eliminate the contagion. So here's our data. I'm going to explain it very succinctly. There's this red line. The red line represents the region above the red line represents where we want to be. So we want to be above the red line. These are all individual data points of people and whether they're PCR negative or not. So we're taking PCR tests. And the red group represents the control. And you just see that one guy by day 7 got above that line. And then you'll see almost, or not almost, everyone was above the line or PCR negative in 7 days. So now let's look at a more condensed summary of the data, and let's look at it versus another drug that we all know very well, Paxlovid. Now, first of all, we can't do an apples to apples comparison, but we're going to show you what their real world data is versus ours. And what I'd like you to do is I'd like you to focus on that green line, and that green line is actually kind of zoomed in on the Paxlovid. So we're going to look at the right side of the slide first. So it takes, look at the bottom, it takes 20 days, see 5, 10, 15, 20 days for the blue line or the blue shaded area to start crossing over into that green area. We want to be above 30% of the people turning PCR negative. So basically, it takes 20 days for you to take Paxlovid for 30% of the population that takes Paxlovid to turn PCR negative. Now look at Bioxytran, and you're going to see the absolute game-changing difference between the two drugs. So we're an oral tablet, and look at that. It's hard to see, but it's 1 day, 3 day, and 7 days on there. And you'll look where the blue line crosses the green line. That's the blue line represents the treated group. It's between 1 to 2 days. Very fast. So we were 88% of the people were PCR negative by day 3, 100% by day 7. Very compelling data. And what was even more compelling about it is we eliminated most of the symptoms by day 7. So we have a repeat of the chart, but on the right was from a journal article, a peer-reviewed journal article. And what you'll see is you'll see the blue represents our drug, the treated group, and that represents their symptoms. And then the orange represents a control. And you'll see in 7 days, where's the blue on the bottom part? The symptoms are gone. There's like no symptoms versus the orange, symptoms went, you got a little bit better, but they're still there. Now, we also have in vitro data that we've done. And this in vitro data shows us, we did it in SARS-CoV-2, that's on the left-hand side. It shows that, hey, we were going to eliminate the virus. So our in vitro data did, and our clinical trial confirmed. So what does the in vitro data say about, on the top part of the right side of the graph is H1N1, which is influenza, and the bottom is RSV. And then what we did is we tried ProLectin-M and ProLectin-I, and you'll see in both of them, we're making a difference. There's definitely some efficacy going on there. So in vitro data confirms that we have a broad spectrum antiviral. And then what else confirms this is literature. There's literature out there that shows that galectin signatures have a broad impact on all these viruses that are on the left and the sources there. So galectins modulate viral infection. I mean, that's what all the literature is saying. And in reality, that's what we're doing with our galectin antagonists. So we think we're going to have profound effect on viruses. And now this is new, and this is a little bit of a shocker here. Take a look at this. There was a case study that was done by some doctors, and it hasn't been published yet. But look what happened in shingles. Here's a before and after. The pain went down really quickly. Look at that, 3 hours. And look at how quick the lesions were reduced by 50%. I mean, you could just see from the area how quickly it went. And then there's another study that doctors have done, and these are with a galectin antagonist. So other doctors have done studies on galectin antagonists and gotten rid of viral conjunctivitis. So now let's look at a ProLectin-I program. ProLectin-I is basically the intravenous version of a galectin antagonist. We are going after long COVID. Long COVID, a lot of people know 1 in 5 people are going to get long COVID. It's affected 22 million people. There's lost work days. There's economic burden. Look at that, 3.7 trillion. This is a problem. And when you think about it, something like chronic fatigue syndrome is very similar to long COVID, costs $9,000 annually to treat that person. So if we came up with a drug, which we think we have, to treat long COVID, that is worth a lot of money. Just take that 22 million people, multiply hopefully less than 9,000, and it's a lot of money. And then 65 to 100 million people are affected in the US, and it often lasts weeks to months. So it's a problem. And I'm double downing on the problem. Look at all these organs that affect: skin, lungs, reproductive organs, head, stomach. Look at all the symptoms. It's a big problem. It's really affecting people's productivity, and there's a couple theories behind it, and we fit into most of these theories actually, but I think the biggest one is the viral persistence. Viral persistence, what have we shown? We've shown that we can neutralize the virus. And then galectin antagonists are also known for helping with organ damage, the microclots, the autoimmune response, and there's even a small case for dysbiosis, which is basically the microbiome in your gut. Now I'm going to look at ProLectin-F. That's basically an intravenous form of our drug that is going to be used for fibrosis, specifically idiopathic pulmonary fibrosis. This affects your ability to breathe. You're looking at age 55 or older. People have idiopathic pulmonary fibrosis. That's where most of the people that get it have it from. We found out that the antifibrotic mechanism is driven by galectin-3, and that's also a universal target for almost all fibrosis. So we think there's going to be a lot of label expansion after we do this. It's an incurable disease, and look at that 3.8-year median survival. And there's 2 drugs out there right now, and they're not that good, and they have side effects. So look at the cost per patient. Direct cost is $190,000. I mean, you're having trouble breathing. What do you do? And then how big is the market? Well, 30 to 40,000 people a year get it. And then that's 20 out of 100,000 people worldwide, according to the NIH. So it's a big marketplace. So now I'm going to talk about the BXT25 and our acellular oxygen carrier and what that is. So let's identify, why do we need an oxygen carrier? Well, we have an epidemic. We call it the brain stroke epidemic. The epidemic is a lot of people are getting strokes. If you look down at the bottom, there's 12 million strokes a year. But now look at the very top, 500 billion in medical costs just in strokes. This is a really big problem. And I got to tell you, we use like the most conservative number out there. It's probably in the billions. And then think about the indirect costs. You'll see the indirect cost over there. These are the caretakers and such. So if we could do something to eliminate this, we really got something. And this is the real upside in our business if we have the funding to pursue it. So the next thing that we talk about is the golden hour dilemma. So what you get a stroke. You can see it. You physically see a person getting a stroke. So you get the onset of symptoms. You call 911. They arrive. So there's a graphic at the bottom. Follow along. 15 minutes later, they're like, "Oh, sir, you're having a stroke." And then what has to happen is there's two types of strokes. One where you have a clot, and one where the blood vessel burst. Well, there's this treatment called TPA, and you can give that to the person, but it only works on the one with the clot, stroke with the clot. But if you give that to a person who has a hemorrhagic stroke, you will kill the person. So what needs to happen is we need to arrive at the hospital. You got to wait a couple hours to get the imaging done to confirm that it's an ischemic stroke and that you can use this. And then so the time to needle is 2.5 hours. Now, while you're doing this, your brain isn't getting oxygen. It's being deprived of oxygen. It's holding its breath. And holding your breath right now, it's not easy to do. So think about the impact it's going to have on the brain. And we've been able to quantify that. And you'll see 2.5 hours is worth about 9 years of aging in terms of how many brain cells die. Now, how do we make the drug? We make actually, I'll tell you how we make the drug, but I want to talk to you about the solution. The solution is this oxygen bridge. So the idea is the paramedic comes, they see the person, "Oh, you're having a stroke." They infuse our molecule, and then all of a sudden, the person's symptoms start to go away, like hopefully right away. And then, and then the person's thinking, "Well, I feel fine. I don't need to go to the hospital." No, sir, you're having a stroke. You need to go to the hospital. We need still to figure out if it's an ischemic or hemorrhagic stroke. But that's what we hope to achieve. And that's how big this could be. Now, the molecule is 5,000 times smaller than a red blood cell. So this imagery shows a clot, and how the blue, which represents our drug, just goes right through it. Now, that's not 5,000 times smaller. You can kind of see. Otherwise, you wouldn't be able to see it on the graphic. But it goes right through. It's almost like in solution with the blood plasma, and it will keep your body oxygenated. And what happens is we have the blood cell, we pulverize it, and we get out the heme, and then we stabilize this heme, which is basically iron. And what it does is it just circulates through your lungs. So it stays in your vasculature, means your circulatory system, and it goes, picks up oxygen, does its thing, comes back 3 minutes, picks up another, and just does it over and over again until eventually the body cycles it out. So that's our acellular oxygen carrier. And I know I'm kind of running low on time, so I'm going to kind of wrap it up here with our investment summary. Look, I'm trying to appeal to the investors out there. Look, when you look at a biotech, the number one thing you're going to look at is, do they have an experienced management team? Look, we've demonstrated with how quickly we've gotten through the clinical trials our expertise. We've done it in record time, cost-efficient development. That's the other thing. We're not wasting your investor money on doing all this research. The research has been done for us. I showed you that slide that shows you how the galectin signatures are on all the viruses. It's done. We know what to do. We just need to test it. So the science has been de-risked. What does that mean? That means we're pretty darn sure this works. What we've done, what no one else has done, is we've eliminated the virus very, very quickly. And it's in a peer-reviewed journal article. And we have the approval to proceed in 2 indications. Now, we're at a value inflection point. And the value inflection point, when we complete our dose optimization trial, and we have that funding in place, which we're very close to doing. The next thing that you can look at is why you should look at our company is we have institutional investors and a very loyal shareholder base. We have a new institutional investor, and his money or their money should help us get to our next regulatory milestone, which is the start of a phase 3 trial in India. And then finally, the reason you should look at us, we have a massive licensing platform. So our idea is never to sell a drug, but to license, license, and license. And that pretty much concludes my presentation, and I can open it up for Q&A.
Great job, Mike. I think we only have time for one, but let's see what we can do. Can you explain? You're good. It's a lot of information. Can you explain today's filings of the S1A? You mentioned that.
Yeah, a lot of people get confused on what these S1s and S1As. S1A stands for S1 Amendment. And we registered some shares. And if you look at our history, it's the third amendment since we filed in mid-June. And that honestly, what that means is that we're going back and forth with the SEC to get our notice of effectiveness. And once we get the notice of effectiveness, it clears the way for us to bring in $500,000 of funding from an institutional fund. And just to refresh investors' memories, that was an above market, or that is an above market deal. We're looking at selling a little over 1.6 million shares for $500,000. It roughly works out to around 31 cents a share. So when you look at the share price of where we're at now, and you look at what an institutional investor said they're willing to put their money in at, it's a significant premium to today's price.
And Cecilia Davila wants you to clarify, you're not a COVID company. So what is the target ailment for your first release?
Oh, we are going after COVID, but the thing that for people to recognize is what our vision is. Our vision is we are going to have that broad spectrum antiviral that eventually a person comes in with an upper respiratory tract infection. So you could say we're an upper respiratory tract infection company, right? Because what our ultimate goal is, you come in with an upper respiratory tract infection, you don't do any tests, or the doctors say, oh, you have an upper respiratory tract infection. Here, take ProLectin-M, go home, you start feeling better, and you'll stop infecting people. And then, because if you have an upper respiratory tract infection, it could be RSV, it could be the influenza, or it could be COVID. And hopefully we get to the point where we prove our case where it doesn't matter what you have, you just take this and you go home. Well, Mike, so we're an upper respiratory tract infection company. Okay.
Well, that was good to clarify. We've got more questions for you, but we do need to move on to our next presenter. But please come back, give us some more updates, and congratulations on your success. All right. We'll see you again soon. Stay with us, everyone. We'll be right back.